Hi, everyone. Thanks for coming to our session today, Thinking Beyond LUCA, STEM Life and Primordial Diversity. So we're going to begin our session today with a remote lecture by Antonio Lazcano, The Last Universal Common Ancestor, What's in a Name? Thanks, Antonio.
My pleasure. I think you can hear me without any problem, right?
Yes, we can hear you loud and clear.
Lovely. Thank you so much. Well, first of all, I want to thank the organizers of this session, Anton, Greg, Aaron, for inviting me. And I have to apologize for being unable to be there. I had a small health issue, so I was unable to travel to Atlanta, which is a place I feel very much at home. And let me start by recalling what I think is an extraordinary example of scientific insight. In 1969, when he got the position of associate professor of microbiology in Illinois, Carl Woe sent a letter to Francis Crick expressing his idea of using different molecular markers in order to study the very early stages of biological evolution, of cell evolution. And if you go down the first page of his lecture, he says something that is extraordinary in terms of his understanding of the basic biological processes of contemporary cells. And he says, well, if I want to study from a molecular perspective molecules, the obvious choice of molecule here lies in the components of the translation apparatus. And he was very right. And then afterwards, after a few years, with the extraordinary collaboration of George Fox, Mitch Sogging, and others, they came out in a classical paper published in 1977 in the Journal of Molecular Evolution. They came out with the idea that, in fact, in terms of a very early divergence, cells could be classified or could be located into one of three major groups. The archaea, until then not first recognized as a monophyletic group, the bacteria, which includes all the prokaryotes we are familiar with, and the nucleocytoplasm of the eukaryotes. And because of the differences in translation and replication between these different groups, they came to the conclusion that these three lines were derived from what they call the progenode. And they gave a very precise definition of the progenode. It was defined originally as a hypothetical biological entity ancestral to the three cell lineages, and that with, they say, a rudimentary, imprecise linkage between the genotype and phenotype. It was a system in which the separation between genotype and phenotype was not complete. At the time, I was traveling quite frequently to Houston to collaborate with John Orro in some issues related to prebiotic chemistry. I had the chance to meet and befriend George Fox, and we actually published in a book in 1992 what we thought was the first and most complete with the information available of the genomic traits of the genetic traits of the progenode. We actually made a very, very detailed analysis of the available literature, and we came to the conclusion that, in fact, the homology between the different components of the RNA polymerase of the three groups, of the elongation factors of the ribosomal RNA, and so on, indicated that translation was of a modern type. We found evidence in the three groups of because of the reports that had been published of DNA topoisomerase 2 of corinode biosynthesis, and this led us to conclude that it was already endowed with a DNA genome. There was clearly an F-type multi-subunit ATPase found in the three lines, and a number of enzymes associated with very basic metabolic processes. So our conclusion was that, in fact, the progenode was not such. It was already a cell very much like the ones that we see today. Now, with the explosion of genomic instruments, with the capability of sequencing very rapidly genes and genomes, and with the availability of more rapid computers, in fact, the attempts to define the so-called progenode, or the ancestor of the three main cell domains, multiplied and exploded in an extraordinary way. And you can see in this list how the ideas, the descriptions of the nature of the progenode diverge. Walsh and Fox had proposed that it was an entity in which phenotype and genotype had not yet occurred. Myself, together with George Fox and John Oro, proposed that it was a bacteria-like cellular entity. Mushegian and Koenig proposed that it was a cell endowed with an RNA genome, an idea that has in fact has been repeated by others like Patrick Forterre and so on. But what is very surprising is the strong differences in our descriptions of the progenode. Some say that it had like membranes. Some say that it was a network of inorganic compartments harboring a mix of virus-like genetic elements. Some say that, like Patrick Forterre, that it was formed by RNA cells that acquired DNA genomes through polyphyletic viral infections and so on. Well, when one looks at the methodologies behind these descriptions, the first thing that is surprising is to realize that there is a confusion between cellular sen ancestors and organismal sen ancestors, which are not quite the same. Every contemporary biomolecule has its own history that traces back to an individual molecular sen ancestor. And this confusion, of course, leads to a very complicated, antagonistic definitions of what the progenode means or what the less common ancestor means. And clearly, something that has to be kept in mind is the reconstruction of the gene complement of this LUCA or less common ancestor must include traits that originated in different evolutionary stages in different cells. Now, clearly, the biases that we have when trying to describe the nature of the less common ancestor have to do with unjustified theoretical assumptions. For instance, the idea that you can extrapolate molecular phylogenies back into the waters of the primitive soup, the confusion between ancient and primitive, people that deny with some people say with very good reasons, I don't think this is the case, the existence of an RNA protein world, with very unrealistic definitions of primitive environments that include some misunderstandings of prebiotic chemistry. And then we have methodological issues like the definitions of sequence usability, biased sample size, primary structure-based searches, undetected gene gain and loss, including, of course, unrecognized lateral gene transfer. But perhaps the most important thing that we have to keep in mind is that nowadays the nature of the less common ancestor should be defined as the nature of the ancestor, the less common ancestor of prokaryotes of bacteria on the one hand, and the archaea with the nucleocytoplasm as a sister group of bacteria. And we have been trying to, in my group in Mexico, we have been trying to characterize the traits of these less common ancestor. And so far what we have is that there is a significant numbers of RNA-related protein architectures. Of course, the obvious are proteins associated with the ribosome and so on, but also many, many proteins associated with the metabolism of RNA. There apparently was a modern type of transcription and translation, a DNA genome encoding a wide array of repair and recombination mechanisms, reverse gyrase, which in the opinion of some colleagues implies a thermophilic lifestyle, lipidic membranes, although the actual nature of those membranes remains an open problem for chemiosmotic coupling, a number of enzymes that indicate metabolic pathways and so on. But what is fascinating is to realize that when you look at the enzymes of some metabolic pathways, you actually see that we fail to recognize, and this is a very common problem, we fail to recognize when you are comparing sequences, many biosynthetic enzymes. And some colleagues have attempted to explain this absence by assuming that the metabolic pathway intermediates were provided by abiotic biogeochemical processes by biosynthesis, prebiotic synthesis that took place in the primitive oceans, that or perhaps that early metabolic routes depended on spontaneous reactions that we know can occur in some cases, or that different organisms were encoding different catalysts that were shared by multidirectional lateral gene transfer. Well, the problem with some of these hypotheses is that if you look at the biosynthesis, let us say, of nucleotides, what you find is a number of molecules that are extremely unstable, and it's very difficult to account for their presence in the primitive oceans of the Earth, or the primitive soup, or the primitive environment, whatever you want to describe it. And I think that one can say that, yes, very likely there were some semi-enzymatic routes that existed during the early stages of cell evolution. This is an argument that Stanley Miller and I insisted on. But in addition, one has to say that we don't have any good prebiotic synthesis of most metabolic intermediates. Most of them are actually quite unstable chemically and have very, very small half-life, and that bottleneck effects will hinder the efficiency of these semi-enzymatic biosynthetic routes. So the conclusion is that most likely we don't have to appeal to these mechanisms that are very, very unrealistic biochemically, chemically, geochemically, and most likely we have been failing so far to identify the missing enzymes. Now, if we go to the issue of the nature of the genome of the less common ancestor, all the evidences that we have collected suggest that there was a DNA genome that was including a wide array of repair and recombination mechanisms. And let me just summarize this, the list, the inventory that we have been able to complete. We have evidence of DNA repair by reversal of damage. We found very good evidence for the conservation of photolysis, for photoproduct lysis. We found very good evidence of excision repair, like uracil DNA glycosylase, a number of glycosylases, excision repair, and so on. And we found evidence of endonucleases associated with UV damage repair. Now, of course, this is not considering the issue of the possibility of lateral gene transfer. But the question is, what was the nature of the less common ancestor? We sincerely believe that the conservation of so many sequences related to DNA metabolisms and the sequence similarity shared by many ancient proteins suggests that there was considerable fidelity already in the genetic system of the less common ancestor. But this similarity is rarely seen. We know that very clearly from RNA genomes, such as viral genomes. It's rarely conserved in the case of sequences encoded by RNA. And well, the question has been being raised back and forth a number of times. And in a very interesting paper published some 15 years ago, Poole and Logan raised the possibility that the less common ancestor was in fact endowed with an RNA genome, since there was, for instance, experimental data demonstrating that viral RNA genomes can undergo repair by the so-called ALK-V mechanisms. And the idea of a less common ancestor with an RNA genome is consistent with Wose's proposals that claimed that the less common ancestor was in fact a highly diverse population of metabolic complementary cellular progenomes with small linear RNA genomes. So is the first possibility, this one suggested, mentioned by Poole and Logan, correct? Well, we do know that in fact DNA can be damaged by alkylation. The bases can undergo methylation. These are very well described, very well understood. And we know of several mechanisms that are present in DNA repair of methylation, which include the cutting off the nucleotide that has been methylated, just cutting off the base that has been methylated, or we know the direct repair mechanisms that recognize the alkylated bases, which is exactly the mechanism that was proposed by Poole and Logan. Well, let us take a brief look at these DNA repair mechanisms that recognize the alkylated bases. And yes, indeed, we have an enzyme that's the ALK-V, which is a monomeric dioxygenase. The enzyme is very well characterized. It requires iron, but the problem is that the enzymes absolutely depends strongly on free oxygen. And clearly, this oxygen was unavailable in the prebiotic and in the primitive environment when the less common ancestor population was floating around. And although it is true that RNA can be methylated, RNA can be repaired by ALK-B, most likely what we are seeing is a lack of absolute substrate specificity, by DNA and RNA associated proteins, as was described in the 1970s by Emil Zuckerkandl and others. So what are our conclusions and our questions? Well, what we call the less common ancestor was most likely a population of complex bacterial-like cells endowed with many of the traits of extant prokaryotes. It was neither a progenitor nor a direct immediate descendant of the RNA world, if there was an RNA world. As of today, comparative genomics is an essential tool, but it doesn't provide us with any direct evidence of the RNA world, pre-RNA world, or much less on the origin of life itself. That I sincerely believe the reconstruction of the trace of the less common ancestor requires a clear understanding of primitive environment and of chemical realities, that the origin and early diversification of life proceeded at a surprisingly rapid pace, and that after what is very surprising is this is something that Stanley Miller and I pointed out many, many years ago. After the rapid biological evolution that took place following the origins of life, the basic genetic trace and major molecular processes have persisted essentially unchanged for more than 3.5, 10 to the 9 years, which I think is quite an extraordinary feat of biological conservation. Thank you so much.
Thanks, Antonio. And we have a lot of time for questions. So I don't see any in the chat right now, but please come up to the mics and ask your questions. We have about 15 minutes for a conversation.
Did I speak so rapidly? I'm surprised.
Maybe I can start with a question. Okay.
Hi, nice talk. Thanks. This is Jen Glass from Georgia Tech. I'm sorry I missed it. Could you go back and I was wondering about your oxygen story. Could you just explain? Okay.
Yeah.
Okay, so how did it repair without oxygen? Can you just go through that one more time for me, if you wouldn't mind?
Okay.
And are there any alternative theories? Yeah, thanks.
Yeah, no, I'll be absolutely delighted. Well, DNA, the bases, and the same is true of RNA bases, can be methylated. And you don't want your bases to be methylated because, of course, you're altering your genetic information. So cells have evolved mechanisms to actually cut off the alkyl groups that can affect the bases. And these mechanisms include the nucleotide excision repair. I mean, you cut off the entire nucleotide and then repair it with a complementary strand. You can just cut off the base and then that has been methylated and then the same thing happens. Or you can actually have mechanisms that recognize directly the alkylated bases, which is the mechanisms that Poole and Logan were suggesting could have play a role in RNA genomes. Well, the problem is that the enzyme is extremely well known. The domain has been, the active domain of this, the dioxygenase has been very, very well characterized. The biochemistry is known in exquisite detail, and you require an iron 2, you require, actually, this is better. You require also glutarate, which is then reduced to succinate. And but the problem is that this enzyme necessarily cannot work in the absence of free oxygen. It's a must. There have been attempts to actually substitute it by nitrogen 2 or other components. It simply doesn't work. So I think, yes, this enzyme can repair methylated RNA, but only under oxygen-rich conditions, which are extremely unlikely in the very early stages when prior to the separation of the archaea and the bacteria. In other words, this is a mechanism.
We have one question in chat, and then we will go back to people here. So a question from chat. Hello?
Louder. Closer to the. Sorry. Okay, this mic's a little better. So we have one question from the chat. How do the two types of membrane lipids fit into the LUCA problem? Were both archaeal and bacterial eukaryote types present from the beginning?
It's a major unresolved issue. People like Julie Pereto and others have argued that perhaps the two types of lipids were already present in the last common ancestor. We really do not know. We don't have a good answer for that question. And that, as argued by Pereto and others, it could be that a different evolutionary trait selected for the separation between the two. But I would say that that's one of the most troubling issues that we actually have when trying to reconstruct the gene complement of the last common ancestor, which is exactly what I have been concentrating together with my group on different traits of possible traits of the LCA.
Hello, Tony Bernetti here from Georgia Tech, and I was wondering, we have this, we developed now this notion that the last universal common ancestor has lots of complicated things, just like a lot of modern cells. I'm wondering what you think of the relative merits of sort of two different models of why LUCA is who and when it is. Like, you can have a model where just attrition over time, you accidentally kill off lineages, versus a model in which shortly before it you have some kind of an explosive advantage. And that could be the reason that the node is where it is. What do you think of the relative merits of these two ideas, or maybe something else I haven't thought of?
Well, no, what I really think is that so far, our reconstructions of the last common ancestor are by necessity based on sequence comparisons, which is fine. But the problem is that a sequence by itself is, well, a set of sequences is not telling you the whole story, the whole biology of the system. What I think is that, and I may be completely wrong in this, but I think is that we had cells populations that were by accretion of genes, by gene duplications, and so on, were undergoing very, very rapid evolution, and rapid relative to geological times, not necessarily rapidly to the rates of evolution of microbes, of prokaryotes. And then one line, by still unknown reasons, was the one that became more successful than the others. But clearly, since lateral gene transfer is a phenomenon that you cannot completely cut off, some of the genes of those other coexisting populations may have survived and may have made it to the LCA. I don't know if I'm answering properly your question, but I think that one simply has to realize how quickly prokaryotes adapt to environmental insults to consider that something equivalent was taking place very, very early on.
Thanks. You've answered my question by saying that it's the wrong question, which is a good answer. Thanks.
Thank you. No, but it's a stimulating question.
Hello, my name is Sausan from the University of Arizona, and I was wondering how we can use all of this information about LUCA's nature to infer the environment it was in. Specifically, that in the introduction you mentioned about how people, I think it was Weiss et al. That inferred reverse sky rays being traced back to LUCA, and then a couple of years later, Catchpole and Forterre were like, no, it was probably horizontally gene transferred from an archaeal domain to the bacterial domain. And that just put the whole thermophilic LUCA debate even on more high stance. And then you mentioned that it might have UV radiation repair mechanisms. So that means it could have been closer to the surface and maybe away from hydrothermal vents, as a lot of people do suggest. And I would like your opinion on how we can reconcile all of this into where LUCA or the population of LUCA might have existed.
Well, the problem is that to reconcile the descriptions of the LUCA or LCA, whatever you want to call it, with our reconstructions of the primitive environment, we will need more detailed reconstructions of the primitive environment, which we absolutely lack. I mean, even today, we have an argument of how abundant was CO2 in the primitive atmosphere. I think we know for sure that there was the flux of UV light was higher than it is today, and hence one can understand that way the ability to repair the presence, the universal presence of UV light damage repair. But I think that the main problem there is that we don't have the precise description of the last common ancestor. There are some things that we know for sure. For instance, a higher flux of UV light. I think that the same would be true for the absence of oxygen, especially prior to the divergence of the into archaea and bacteria. But other than that, I will be very, very skeptical of making very strong statements, although I have to say that I do like the idea of LCA or LUCA living in lukewarm waters. Is this contradictory with the idea of a hot origin of life? No, because LUCA is very far removed from the origins of life, if not in time, at least in complexity.
So we have a couple of short questions from chat. First one is from Fabia Ursula Batutsi. How widespread is the gene ALK-B in prokaryotes today? Are there any alternatives to perform the same type of repair?
It's a very good question. The gene is extremely widely distributed. It's found also, of course, it's present in aerobic prokaryotes. It's extremely abundant in ALK-B. It's absent, and that's easy to understand, in a number of parasitic eukaryotes that live in the guts, for instance. And the homologs of the ALK-B domain have a very wide distribution.
Brief question.
Thank you very much for your presentation. My name is Vladimir Subotin, University of Wisconsin. You already partially answered my question that it's the greatest mystery that information molecule, RNA or DNA, appeared in a form bacteria-like ancestral structure already surrounded by membrane. And how membrane simultaneously occurred together with information molecules is a great mystery. You said it. But could you imagine evolution of membrane compartment themselves, like in a lipid world hypothesis, but as a sphere or in a form of liposomes? That we know that liposomes can divide, they can fuse, and they can be in a different level of a water pool, on a bottom, on a top, and they can actually evolve themselves without any information molecules. Have you ever considered this hypothesis?
And you're raising out what I think is a fascinating issue. I don't think that mechanism will apply for the less common ancestor for the LUCA, but certainly, one thing that biologists have not considered sufficiently is what is called a structural heredity, which clearly is involved in the multiplication of structures in the absence of genetic material. A good case will be the centromeres or centrioles. Another case will be, in fact, what Leslie Orgel termed replication with no transmission of hereditary information, which will be exactly the case of the liposomes. I think this is not denying the key role that genetic material has in evolution, in continuity in time, and opening the possibility of divergence and adaptation and evolution and adaptation. But clearly, it's a mechanism that I would certainly not put away when thinking of precellular systems.
Yeah, and the next question. Do you think that heredity of liposomes could be present as a content of liposomes?
Sorry,
Not
we are running out of time, so we just had time for one question. So I would just want to thank Professor Lascano for his wonderful presentation and his answers to all these many questions. But we have to move to the next presenter, who is Timothy Sokolsky, and who is going to present a talk on origins of the modern reductive tricarboxylic acid cycle in the evolution of life. Please.
Thank you.
But if you run out, don't be tough for questions. Got it all in the notes. Got it. Thank you. So it's really easy, okay?
Hello, everyone. My name is Tim. I'm a first-year PhD student at the University of Wisconsin-Madison. Although this work was actually conducted last summer during the Blue Marble Space Institute of Science Young Scientist Program. So the topic of my presentation is the origins of the modern reductive TCA cycle and the evolution of life. Carbon fixation is one of the most fundamental processes in life today. In simple terms, it just involves incorporation of carbon dioxide from the atmosphere into biological molecules, and is present in all three domains of life, in archaea, bacteria, and in eukaryotes. And there are actually many different biochemical pathways with which life can fix carbon, some of which you might have heard more about, such as the Calvin cycle, which is common in cyanobacteria and plants, or the reductive acetyl-CoA pathway, also known as Wood-Lindahl pathway, which is this ancient carbon fixation pathway present in some methanogens. But of the main interest to our study was the reductive TCA cycle, reductive tricarboxylic acid cycle, which is basically Krebs cycle that runs in reverse. So Krebs cycle is the oxidative TCA cycle. It runs in the oxidative direction and releases CO2, and reductive TCA fixates CO2 instead. So RTCA is a very important biochemical pathway for the field of origins of life and astrobiology in general, for a few reasons. First of all, RTCA is autocatalytic, and autocatalytic pathways are particularly interesting because they present a really unique way to rapidly increase complexity in the prebiotic setting. And also, RTCA involves biologically common metabolites, so tricarboxylic acids that are present in basically all organisms. And there were studies that have shown that some reactions of RTCA can be catalyzed without the presence of enzymes, for example, using minerals. But of the main interest to our study was that RTCA has a really strange and patchy phylogenetic distribution. It is present only in a few rare groups of bacteria, and that's it. No archaea, no eukaryotes, only a few rare groups of bacteria, which is not exactly very consistent with the idea of it being this really ancient carbon fixation pathway. So here's what the actual cycle looks like. First of all, you can see that 2 oxalacetates are produced per 1 round of the cycle, and this is the autocatalytic part of it. And so 1 oxalacetate is coming from pyruvate carboxylation, and the other one from the cleavage of citrate. And also you can see the 3 irreversible steps of the cycle that are catalyzed differently from their counterparts in oxidative TCA. So 2 of them are marked in red here, and the 3rd one is actually this reaction, which is the reaction of citrate cleavage to oxalacetate and acetyl-CoA, which is also key because it enables the autocatalytic part of the RTCA cycle to work. And there are actually 2 distinct ways citrate can be cleaved. The first way is just a simple 1 enzyme reaction using the enzyme ACL, or ATP citrate lyase. We just cleave citrate into acetyl-CoA and oxalacetate. And the second way is actually when citrate is first converted into a citral-CoA intermediate using CCS, or citral-CoA synthase. And then citral-CoA is cleaved by citral-CoA lyase into acetyl-CoA and oxaloacetate. And here you can also see the reverse reaction of CS, or citrate synthase, that catalyzes, well, the synthesis of citrate, which is just a simple one-step process. So here is a pattern of how all the enzymes that I just mentioned, with a lot of abbreviations, are related to each other. Some of this is from the literature, and our findings also confirm this scheme. So first of all, you can see the two-enzyme system, which is often proposed to be ancestral. So you have the enzyme CCS that produces citral-CoA, citral-CoA synthase, and CCL, which cleaves citrate-CoA, and is present in only a few rare groups of bacteria. Also, these two enzymes, CCS and CCL, have homologs among other TCA enzymes. For example, CCS is homologous to SCS, or succinyl-CoA synthase, which catalyzes succinyl-CoA synthesis, or breakdown, in both oxidative and reductive TCA. And then CCL is homologous to citrate synthase, which is an enzyme exclusively unique to oxidative TCA that catalyzes citrate synthesis. And subsequently, so also looking at homologs here is especially important because they're much more common than the actual citrate cleavage enzymes and did not experience as many losses in their evolutionary history, so we can infer a lot more from looking at their evolution. And the last citrate cleavage system is the more derived one enzyme system. That is just ACL, that first started out as a 2-subunit enzyme that still catalyzed the one-step reaction without the citral-CoA intermediate. And this resulted from fusion of one of the subunits of CCS and CCL. So this is the intermediary step, basically. And then the two subunits of ICL fused again in eukaryotes. And you get just one subunit enzyme that does the same thing. So in order to investigate the rarity of this key reaction, we tried to do a survey of its distribution and phylogenetic analysis using various databases such as KEGG, NCBI GenBank, or Syntax. So first of all, what we found is that the distribution, so here are some of our findings regarding the distribution of a citrate cleavage reaction overlaid on the tree of life. And you can see a few things here. So first of all, we identified a few taxa in which complete RTCA is theoretically possible. At least they have all the necessary enzymes for that. And note that there is only one archaeon, the microarchaeota. And it's basically the only archaeon we found that could even theoretically have RTCA. But the main finding is that from this, it's really unclear which of the ancestors had which enzyme and how this whole system evolved as a whole. But what is interesting is that most taxa that had the two-enzyme system, so CCS and CCL, actually were oxygen-tolerant thermophiles with incomplete TCA, so incomplete either oxidative or reductive TCA. And what's particularly interesting is oxygen tolerance, defined here as both aerobes and microaerophiles, because it's really strange that the ancestral form of this supposedly ancient reaction is actually mostly present in oxygen-tolerant organisms. Then we looked at the phylogenies, and I believe I don't have enough time to get into too much detail with these, but the main takeaway is that, as you can see, there are very few monophyletic taxonomic groups here, which indicates that there were likely abundant horizontal gene transfers in the evolutionary history of these enzymes. So this is the phylogeny of ACL, the one enzyme system, and this is the phylogeny of CCL, part citral-CoA lyase, and its homolog citrate synthase. With succinyl-CoA synthase, though, it's a little bit more interesting, because what we see is that multiple distantly related citrate-cleaving clades actually have monophyletic SCS. So this implies either that succinyl-CoA synthase is capable of citral-CoA synthesis as well, which was empirically demonstrated to not be the case, at least with a few citral-CoA synthases that we know of empirically. And therefore, the likely conclusion is that it was due to horizontal gene transfer event or a sequence of events from a similar source organism. And also adding to that, as we found by looking at synteny for all these organisms, TCA-related genes are often located in close proximity to each other in the genomes, which increases the likelihood of some kinds of linked transfers, linked horizontal gene transfer events, which in general implies that citric cleavage might have been also obtained horizontally, linked to SCS in these organisms, in these bacteria specifically. Again, we don't see this pattern in archaea that have citric cleavage, but don't have complete RTCA. Albeit it is important to mention that deep time horizontal gene transfers are next to impossible to definitively resolve, particularly since parametric methods of inferring AGTs, such as comparing GC content or cotton bias, just don't work here because we're talking about events that could have happened billions of years ago. So this is basically all we have. Another interesting implication of our findings are in regards to the evolution of metabolic mutualism, or syntrophy, especially in the case of RTCA. So let's look at this example first. An established ectosymbiotic relationship between nanohalarchaea and halarchaea, both groups of archaea that inhabit highly saline environments. And what we see is really interesting. So halarchaea, a really well-known and well-established group, have basically all RTCA enzymes with the exception of one, or one enzyme system, the system for citric cleavage. While nanohalarchaea, which are frequently known to be their ectosymbionts, are known to only have citric cleavage enzymes, particularly of the two enzyme system of CCS and CCL, which implies, of course, a possibility that there could be some kind of syntrophy going on, with citrate being transferred from one archaeon to another, and the product, like oxalactate or acetyl-CoA, being transferred back. And there are actually multiple suitable transporters in both organisms. And this is obviously speculative and requires empirical validation, but we see a very similar pattern for a lot of other groups of syntrophic partners in our study, such as asgararchaea and mesenogens, or microarchaeotes and ceramoplasmas, and a few other groups as well. So this is not just unique to halarchaea, although yes, it does require empirical validation. But why it's interesting specifically with halarchaea is because it actually adds more context to one previously proposed idea, or the purple Earth hypothesis, which basically states that due to spectral complementarity of halarchaeal bacteria rhodopsins, so marked here, and chlorophylls, the evolution of the latter could have happened due to the competition for wavelengths with retinal phototrophs, so like halarchaea, again, that use bacteria rhodopsins, which have a retinal pigment. But this is complicated by the fact that there is no carbon fixation in modern halarchaea, so there's no coupling of a process that produces a lot of reducing equivalents, like photosynthesis, to a process that requires them, like carbon fixation. And our findings provide a possible explanation for that, either through possible RTC in the past, and therefore citric cleavage enzymes in nanohalarchaea being acquired horizontally, or just through RTC operating syntrophically. And we also discussed a little bit about how syntrophy could be in general important for the field of origins of life, because it's significantly more common than expected, and was shown to be product of selection rather than random losses. And there are even a few lines of evidence for the prevalence of syntrophic interactions early in the evolution of life as well. So basically, our main conclusions are that citric cleavage in modern bacteria possessing RTCA is likely a consequence of deep time horizontal gene transfers, and therefore modern distribution of RTCA might not reflect its origins, and that syntrophic interactions might have played a role in the evolution of RTCA, origins of life as a whole as well. And as I mentioned, this project was started during the MSIS Young Scientist Program, and I would like to thank the whole the Sarma Lab for all the help and feedback on this project. Thank you.
Thank you. We have a time for one short question.
Hi. Oh, God. That was a great talk. Julie Huber from Woodsole Oceanographic. And I just want to say this is why I like coming to these meetings, because your rare, organisms are the most abundant organisms at deep sea hydrothermal vents. And I was curious, when we sequence these environments, over half of them are epsilons and aquaficales. And I was wondering if any of the metagenome assembled genomes that we're pulling out of these natural ecosystems that represent way more than the culture diversity, if they can be incorporated into your work.
Thank you for your question. Yes, I was trying to largely avoid environmental assemblies just because a lot of it is uncultured, and it really pains me every time I see a good enzyme that is just from something that's not very well known. But yeah, I think I only incorporated a few environmental assemblies just from a more well-known taxa, at least from the literature perspective. But yeah, RTCA could definitely be significantly more distributed. And actually, one of the ideas that was previously proposed as well was that it originated in archaea instead of bacteria, even though it's now entirely absent in archaea, which could potentially suggest that there could have been some kind of lineage that we either don't know about or that just died out at some point that possessed this cycle and then transferred it to modern bacteria. So yeah, looking at environmental metagenomes is definitely really interesting, especially for this kind of project.
Great, thanks.
Do you have more questions? Or no? Okay.
Thank you, Kim. And we'd like to move into the next speaker, who is Veronika Mierzejewski, and she is going to be talking about scaling patterns in enzyme diversity and the universality of life. Please.
All right, hello, my name is Veronica, and I am going to talk to you all about a new way of finding universality in life. To start, we're all familiar with the fact that life is very diverse on Earth, and apart from a shared genetic code, we don't actually know what is common to all instances of life. So that's a question I'm really interested in. What unites all life in the universe regardless of environment? This is a useful question just because it's interesting in itself, but also uncovering universal features of life will help us in endeavors such as constructing minimal life, constraining early models of the earliest life, and searching for life agnostically, or weird life. So one way to find this universality is to look at scaling patterns. And if you're not familiar with scaling patterns, all they are how some attribute changes as the system size increases. So you can have a linear relationship, but you can also have other interesting ones, such as sublinear or superlinear behavior. And throughout this presentation, I will demonstrate scaling relationships in the way I just showed you, as well as in log space, where the slope denotes what scaling behavior is. So above 1 is superlinear, below 1 is sub, 1 is linear. The good news is that we've found scaling in biology already. So a very common example is the metabolic rate of animals, where on this plot in the x-axis you see body mass, and on the y you see metabolic rate. And I wanted to stress that it's very, very strange that so many of these diverse organisms fall within this line. And what it's showing us is that with all these organisms, despite their differences, are all experiencing the same universal constraints. So the reason we see this, we learned decades after this observation, is that there is a universality in cardiovascular branching patterns that's optimized for energy transport. In case you're not convinced that scaling is very useful, especially in the purposes of astrobiology, a quote from Jeffrey West, who does research in this work, has a wonderful quote laid out. If you tell me the size of a mammal, I can use the scaling laws to tell you almost everything about the average values of its measurable characteristics, such as how much food it needs to eat each day, what its heart rate is, how long it will take to mature, the length and radius of its aorta, how many offspring it will have, and so on. And this is just knowing one piece of information, which is the size, and no other details. So scaling laws have immense predictive power. Now, what I want to do is find hidden universality within biochemistry, because fewer studies have looked at that. So when we talk about size here, what I'm referring to is the size of an organism or community's biochemical space, or where an organism is capable of biochemically. And an attribute, what I'm interested in is in the diversity of biochemical functions. And so what I'm showing here is pretty abstract. And one way to get about this, to make it more concrete, is to look at enzymes, because they're a great proxy for biochemical function. They catalyze most reactions. And along with that, conveniently for us, they're classified by a 4-digit EC code that is related to its function. To break that down, at the first digit, we have the broadest function. For example, EC1 are the oxidoreductase enzymes. And they're any enzyme that transfer electrons from one molecule to another. When we get to the second digit, we're adding more specificity. We're saying that these are oxidoreductases that use CH-OH groups as electron donors. By the third digit, we're adding more information. We're specifying the electron acceptor. And by the time we get to the fourth digit, we're specifying the exact enzyme. So this is useful for us because we can look at different specificity levels of function. To give you a bird's eye view of all of the broad functions, all of the first digit ECs, we have the oxidoreductases, as we just mentioned, transferases, which are enzymes that transfer molecular groups from one molecule to another, hydrolases, which cleave molecular bonds using hydrolysis, Lyases, which cleave bonds using any other method besides hydrolysis. Isomerases, which shuffle molecules. And ligases, which form molecular bonds. So going back to this, rather than saying the size of biochemical space, like what does that actually mean? We can look at the total enzymes that are encoded in a genome or metagenome, because that data is actually available to us. And instead of saying diversity of biochemical functions, we can talk about the number of unique enzymes within a particular EC class. So in the past, we asked if enzyme diversity scales across the domains and across levels of biological organization, aka on the organism scale versus ecosystem scale. And we looked only at the broad first digit ECs for this question. And we also want to know, is there universal behavior for all of these biological groups? Because there's no reason to suppose that there would be such a relationship. The way we did this is we used the Joint Genome Institute, or JGI, which contains thousands of genomes and metagenomes. And they're lovely because they have a pipeline to annotate these genomes and infer what enzymes are present. And from there, they have EC codes tacked onto it. Nice way to organize our data. And with our data set post-filtering to make sure we have high quality data, we have hundreds to thousands of samples, which is enough for us to make some statistical generalizations. So good news, scaling does exist at the broadest first digit EC function, and we showed that in our most recent paper. So what you're looking at here is all of the scaling relationships for the first digit. And if we're looking specifically at one data point, that represents a genome. And on the x-axis, that's the total biochemical toolkit. And on the y-axis, we have how many unique enzymes are within that particular class. And also, there is universality for a lot of these ECs. So for example, with the oxidoreductases, it's super linear whether you're talking about bacteria, archaea, or the metagenome, which represents a large-scale ecosystem. And here what I'm showing are the slopes of the trend lines that I just showed you in the previous plot. And the universality comes from the fact that within a given row, we have the same color across the board. Now you'll notice that with the lyases and the isomerases, they're not demonstrating that universal behavior. And I have some ideas as to why. So with the EC system, that was made for biochemists to do traditional biochemistry, but what we're after is to look at a more abstract sense of function. So if we combine hydrolases and lyases, which are enzymes that both cleave molecular bonds, then we see that universal behavior, and it happens to be superlinear. Isomerases, still not universal, but I suspect that's because those enzymes act on one molecule, and typically you'll have things like intramolecular oxidoreductase reactions in them, which I think makes that more of a mixed bag category. So currently, what I'm after is I want to see if there's scaling behavior for the more specific ECs, the second and third digits. I also want to know if that scaling is universal as you look down at levels of specificity. And finally, I want to see to what extent scaling laws hold in deep time, and I want to see if the scaling coefficients or the slopes are correlated with phylogenetic distances. The idea being that if you're more closely related to one organism, you'd have a similar slope to that organism. So looking at this first question, good news again, scaling does exist at the second and third digit level. So what you're seeing here are oxidoreductases across levels of specificity, and these all happen to demonstrate superlinear behavior. Interesting. The same is true for ligases. These ligases all demonstrate sublinear behavior. Now, is it the case that it's universal across levels of specificity all of the time? Not really. So that is not always the case. For example, with the transferases EC2, that demonstrates sublinear behavior. But when we look at the second digit, then we see more of a mixed bag of different sublinear, linear, and superlinear behavior. Now, finally, is there a correlation between scaling coefficients and phylogenetic distance? What we found when we examined these slopes at the phylum level is that there are differences in these scaling slopes. So what you're looking at here are different phyla of bacteria across these different ECs. And plotted here on the lines are the slopes and their confidence intervals. So you can see there are differences, and not surprisingly, the oxidoreductases have slopes above 1, the transferases typically have slopes below 1, but the point is that they're different. But they're not necessarily correlated with phylogenetic distance. So this is a tree representing bacterial orders. And these colors on the grid plot represent the scaling slope. So you'd expect there to be more similar colors clustered together if there was a correlation. But that's not looking like that's the case. We're going to perform more statistical analyses to make that result more robust. So if it's not correlated, but there are differences, then could it be the case that the random partitioning of taxa into different groups is, what leads to these scaling slopes? In other words, if we randomize what taxon is in which group, will we still see the same thing? The answer is we don't quite know yet, but I do want to illustrate what I mean by this idea. Currently, I'm running these permutation tests, basically mixing up which taxon goes where. And the idea is if the standard deviation of our true data happens to lie outside of the permutated standard deviations, then we can say with some degree of confidence that it's not the result of just random partitioning. And if that's true, then that makes it more interesting because there might be some physical reason at play that we just don't know about. Okay, so summary of all the questions, answers so far. And my future goals are to investigate why there are differences in these scaling slopes. I also want to know what specific biochemical functions drive the first digit scaling, because I think that will give some hint as to what the mechanism is and point more towards why we see this universality in the first place. And I also want to control for sampling bias in the phyla, so it's not uncommon for a given phylum to have thousands of samples, whereas another one might have 50. So in order to make this more robust, I want to make sure that all phyla are evenly sampled. So going back to these goals that I mentioned earlier, I believe that universal scaling in function can aid in these endeavors. And particularly with LUCA, we can infer how many enzymes might have been present in this system. And using these scaling laws, we can actually determine what ratios of functions we would expect. And along with that, for searching for life agnostically, we're talking about function here. And that means that we can substitute compounds, and we can think about alternative biochemistries to make our search for life elsewhere in the universe a little more open-minded. So big thank you to the eLife team and for our collaborators, and thank you all for being here and listening.
Hi, I'm Vladimir Subbotin, University of Wisconsin. When you showed this graph that represent size and metabolism, all animal fall.
Oh, yes.
I wonder that you restricted this representation not only by vertebrates, not only by mammals, but particularly to placental mammals. And what about other taxa? For example, what jellyfish or octopus?
Yeah, for sure. Yeah, so fortunately, other scholars have investigated this question and have looked at all life. So there is a plot that exists that includes things like prokaryotes and non-placental mammals. I just chose this plot for simplicity to illustrate this idea that you can have really disparate organisms that fall within this universal line.
Thank you.
Hi.
Oh, is that it? Okay.
No, you're
Oh,
good.
okay.
We just have some questions in the chat.
Oh.
A question from Joanna Monti Maisel. How does this compare to the scaling laws for genes?
Describe it
Yeah. The chat keeps moving.
by Molina and Van Nimmer. Do I have this correct that you are getting differences among phyla in enzymes, whereas they fail to find them in other gene types such as transcriptional regulators?
I'm sorry, I didn't catch that.
How does this compare to
Hope everyone's awake now. Okay. I don't think this is working anymore. Yeah, that's a really great question. I'm actually not familiar with that particular study, but I will look into that and get back to you if possible. That's really interesting. Because so far we've only looked at genomes and we haven't looked at transcription data yet.
And really quick, one more question from the audience.
Hi, so great talk. Just have a kind of a quick question related to transcription. I know I haven't looked at it yet, but you can kind of do a census of enzymes in presence, but they may not necessarily be part of functional pathways, particularly in some archaean bacteria.
Yeah.
So I'm wondering if you've looked at complete pathways and if the scaling patterns remain in some of those complete pathways rather than just in individual EC classified numbers.
Okay, it was a little bit hard to hear you, but I think what I heard was that we don't necessarily know if those enzymes are expressed. And that is a shortcoming of this study, right? So there is far more metagenomes and genomes available than transcriptomic data. And the transcriptomic data that does exist is, at least in JGI, is usually the metagenomic data. Whereas for this study that I'm currently working on, I'm interested in the more individual organisms like archaea and bacteria. But if you know of data sources that have such data, please let me know.
Thank you.
All right, thank you.
Okay, thanks.
So next we have Tomasz Zaszkowski for an evolutionary perspective on prions, trends and function of prion candidates across all three domains of life.
So at 12 minutes, I'll just gesture and I'll give you 3 minutes for questions, but I'll let you wrap up your thoughts and go into that time. So, all right.
Thank you so much for coming. My name is Tomasz Zajkowski. I'm a neurobiologist fascinated with the early evolution, and I'm working at NASA Ames with Lynn Rothschild. Today, I'd like to share with you a radical idea that came to my mind during my PhD, an idea that an aggregate discovered in human brain could be a relic of early evolution. So in my research, I focus on prions. Prions are fascinating because they're so controversial. Let me give you an example. Prions were first discovered as a protein aggregate in patients suffering from infectious brain disorders. But first, prions were thought to be viruses. I mean, those disorders were thought to be caused by viruses. It took decades to accept the controversial idea that the protein itself could carry biological information from an organism to another, eventually causing a disease. So for the first 20, maybe 30 years, prions were widely recognizable as infectious proteins, but recently we have experienced dramatic shift in perspective on prions, maybe even to the extent that the name itself no longer gives the justice to the phenomenon. The name prion was invented to reflect infectious nature of proteins. It is an acronym for proteinaceous infectious particle. But in recent years, we have found not only prions in mammals, but many other organisms, including bacteria, archaea, and even viruses. So, and since the term infection is reserved for invasion and growth of germs in a body, the name itself no longer properly describes the phenomena. So it was, if that wasn't enough, prions were recently found to play beneficial functions, both in human and microorganisms. In yeast, prions can facilitate rapid adaptation to changing environment, and they are a powerful epigenetic mechanism of adaptation that likely existed more than 1 billion years ago when these two species diverged. In fact, some researchers are so impressed by the new perspective that they call it the largest paradigm shift since identification of DNA as a molecule of heredity. Pretty bold. In a paper that we published last year, we have found that candidate prion proteins from archaea can transmit phenotypes for many generations when expressed in model organisms. The motivation for this paper was to search for prions specifically in the domain archaea because it was the last domain of life not studied in regard to prions at the time. And And we found some. With this work, we have helped to close the gap in understanding how widespread the prion phenomenon is on the evolutionary tree of life. And it was the first step to start asking questions about the origin of prions, so climbing the evolutionary tree of life downward. So could it be that prion aggregation or protein aggregates capable of storing biological information exist on early Earth? Could they be forming abiotically? What roles could these aggregates have in early life? Or maybe could they even facilitate the origin of life? These are the major questions that motivate my research. To understand the potential functions of the oldest prions on Earth, let's spend a little bit of time looking at how they work in contemporary organisms. So on this animation, you can see a prion protein in its native soluble state. And like all prion proteins, it can misfold, another term from medicine. Actually, more adequately, we should say it obtains an alternative fold. It changes its conformation, increasing its beta sheet content. In this form, it can force other copies of itself to do the same. And in this stage, these proteins can adhere to each other, forming a very tight interactions, and in consequence, forming an insoluble amyloid fiber. That's shown on the right. Amyloid fiber is very rigid. It's resistant to high range of temperatures, pH, and even degradation. And importantly, amyloids can be made out of very short peptides, even prebiotically possible. So it's likely that amyloids existed on early Earth. Importantly, amyloid grows exponentially fast. At first, there is a lag phase, but then it grows exponentially quickly, and it quickly leads to depletion of natively folded versions of the protein that builds it. So prion proteins that have amyloid fold in it, these prion proteins aggregates in the form of amyloid are often unable to perform their functions that they had before they aggregated. So if that happens in a microorganism, phenotypically it looks like the gene deletion, but in fact the gene is not affected. Therefore, the change in the phenotype depends on the protein aggregation, and it's shared epigenetically in the cytoplasm. And just like in the case of chromatin epigenetics, the trait can be heritable because the aggregate can be passed to daughter cell and continue recruiting freshly synthesized copies of the same protein to the growing amyloid, rendering the protein unable to perform its functions for many generations. If the amyloid aggregate can be passed to another generation of cells, this is what we call prion. So in this sense, prion is a heritable aggregate or condensate that can change cellular phenotypes for generations. In case of classical yeast prion protein SUP35, the switch to amyloid promoting state happens every 1 million divisions and is stable for 10,000 generations. In other words, prion behave as molecular switches. They can broaden phenotypic diversity. And paraphrasing Adriano Aguzzi, having a protein that can exist in on and off state, where one of the states is heritable, is a wonderful way to transmit information, and it would be surprising if nature didn't utilize it for this system in order to solve specific problems during evolution. And here are some examples how yeast can benefit from prions. On the top left part, you can see the prion GAR+. The rate of GAR+ formation can be influenced by lactic acid, and in consequence enabling yeast to use various carbon sources, switching from metabolic specialist to generalist. Other consequences of prion formation include switching to multicellularity, increasing sporulation, or even expressing subtelomeric regions, which is an example how prion mechanism regulates another epigenetic mechanism. My personally favorite example is prion PSI plus made of SUP35 protein that I mentioned on the previous slide. It's Formation allows to read through stop codons. It uncovers hidden genetic traits. So this can be a wonderful mechanism facilitating evolution because anything after a stop codon is not experiencing selection pressure and can mutate freely. But after the prion is formed, it suddenly puts that polypeptide to test. As I mentioned, I'm very interested in finding out what are the oldest prion functions. So recently, machine learning tools were developed that allow the prediction of prion behavior bioinformatically based on amino acid sequences. So our recent work focused on computation, on bioinformatics, and the future work will again be lab-based prion confirmation of the behavior of the proteins. So the problem with prions is that unfortunately their sequence of the prion domain, the fragment of the protein that is responsible for aggregation, those prion domains are very homogeneous and in many cases consist of just long stretches of glutamine asparagine. This composition of prion domains challenges the conventional phylogenetic approach. It's really hard to say if they are conserved based on sequence. Prion domains are modular. That's another characteristic of prion domains. That means that you can attach it to another non-prion protein and convey the ability to form a heritable aggregate to it. So we use prediction algorithms to identify candidate prion domains in all proteomes of UniProt, from UniProt, proteomes of organisms representing all three domains of life. And then we annotate functional domains of the candidate using gene ontology and KEGG description. So the other domain except the prion domain that exists in this polypeptide. So we analyze the functional domain that coexists with prion domain in the same polypeptide chain. So first we generated phylogenetic trees marking the predicted prions in all available high-quality proteomes. The blue lines on the tree indicate proteome with at least one prion candidate in it. The bars around the tree indicate how many prion candidates per proteome we have found. So blue is more than 5, green is more than 10, and red is more than 15 prion candidates per 1,000 proteins. The take-home message is that eukarya have much more prion candidates per protein than other domains, and it could be that better protein aggregation handling systems in eukaryotes allow them to use prions for more widely avoiding lethal aggregations, but it could also just be a bias of a prediction system that is trained on eukaryotic proteins. So next we looked at enriched functions of prion candidates in different domains. Enriched means that these specific geoterms were more likely to be found in prion in a protein with a prion domain than in a protein that doesn't have a prion domain. And we found 2 enriched functions coexisting with prion domains that are conserved across all 3 domains of life: calcium ion binding and helicases. Calcium ion binding is a common for proteins engaged in signaling, and this observation goes well in line with the hypothesis that prions can work as hubs that integrate diverse inputs and actuate diverse outputs as molecular switches, just like in signaling should do. And helicases can influence activity of many genes at the same time, and this can provide immediate access to genetically complex traits. This role of prions was already suggested in yeast, and our data shows that it could actually be conserved function of prions in all domains of life, and potentially the most ancient prion functions as well. So looking at KEGG annotation of proteins annotated with GO as calcium ion binding, consistently we found the same description in all domains of life, but none of them was yet shown to behave as a prion in experiments. So these are all very interesting candidates to study in the lab. Looking at KEGG annotations of proteins annotated as helicases, we found several confirmed prions. In bacteria, the most common KO annotation was transcription termination factor Rho, and the Rho protein was the first prion confirmed in bacteria, specifically Clostridium botulinum. So in eukaryotes, multiple different helicases showed up in the data set, including DDX 3, 5, and 6. In a very recent publication, our collaborators from Stanford, they showed that DDX 5 forms cytoplasmic aggregates in old fish and mice, and when they express it in yeast, it forms heritable aggregates. So these experiments strengthens our confidence in the prediction model. So let's go beyond the enriched functions now and we have to finish? All right, so that's a conclusion. On the left side, there are numbers of overlapping functions, so there is many more if we're not looking at the enriched ones. So I'd like to finish the presentation with a hypothesis that we came up during analyzing this data, that prion regulation of the fundamental cell processes could be an evolutionary ancient one, even if the sequence of the individual prion domain are not evolutionary conserved. And thank you so much for your attention, and to my coworkers.
Thank you, Tomasz. Great talk. So, unfortunately, we don't have time for questions. But moving along to our final speaker of the session, Fouad Al-Baidouri will be presenting work entitled "The Phenotype of the Last Universal Common Ancestor and Evolution of Complexity." It's going to be like 12 minutes, and then 3 minutes for questions.
I'll do my best. Okay, should they just close this and start? Hello, everyone. Great to be here. Most of the main message, let's say, from today can be summarized in the first conclusion of Antonio Lascano talk. That is, LUCA is probably a complex population of cells that resembles modern prokaryotes. I'm currently a research associate working on malaria at Harvard University and Broad Institute, but let's talk about LUCA. So, as Lascano also has mentioned it, LUCA is way far in time after the first after life originated. That is to say, if we consider the Darwinian threshold hypothesis suggested by Weiss, that is, there is an RNA world, there is then progenote-like up to LUCA. So when talking about LUCA, we are talking about this common ancestor of all living organisms today. Anything related to LUCA reconstruction is subject to debate, obviously. So we all know this image, and we grow up with this, with having a tree domain of life trees with bacteria, archaea, and eukarya. And as you know, the discovery of Asgard archaea and other archaean cells that share many characteristics with eukaryotes led us to think that there is probably just two domains of life. Bacteria and archaea with eukaryotes is a group that emerged much later during evolution. With LUCA and its derived and its descents, like the last bacterial common ancestor and the last archaeal common ancestor, for which also I will show some reconstructions. So obviously, when we come to the reconstruction of ancestral states and go back in time, we usually use fossils. Unfortunately, for microbes, this is not possible. And we therefore rely mainly on the genomes or genetic sequences or any other statistical inference in order to reconstruct past characters. So for a long time, LUCA has been thought as a minimal or primitive cell, like maybe half a life, and recent reconstructions by Weiss et al. Have concluded that LUCA must have been like a very simple cell with about 350 proteins. This is just to give you an idea, is about the size of obligate endosymbiotic bacteria that we find today. So all of the reconstructions that we know of are based on the absence or presence of genes and based on genomic data. Here, I will talk about a different approach that is based on phenotype and not genomes. So we know that it is still inherently difficult to predict phenotype simply by presence or absence of genes, although we can do it in some cases. And here we will use, so if we consider observed traits that we have today, let's say motility or shape or whatever, so we can use, in this example, Bayesian inferences and using Markov models, and we can infer ancestral states and obtain some posterior probabilities for those states. So those ancestral state reconstruction methods has been used to for like diatoms. They has been done also on other eukaryotes, and more recently we used this method to reconstruct ancestral states for Firmicutes bacteria. So to reconstruct the character, the ancient character of LUCA, I have collected more than 3,000 phenotypic characters, and I went during 3.5 years over the Bergey's Manual of Systematic Bacteriology, and we collected data on shape, motility, environmental data, spore formation, membrane, etc. And obviously, when we reconstruct ancestral states, there is the problem of horizontal gene transfer. In our case, what we did is we did some simulations on horizontal trait transfer. That is, considering a trait and how it can be transferred and how this transfer might impact our constructions. And just to summarize here what I want to show you, we did two different simulations, one that is a local transfer approach, and the second one is a global transfer approach. And we expect that using a Brownian motion model, and we expect that the slope will, in a case and that it doesn't impact our construction, it will be 1. And as you can see, the more we increase the number of simulation up to 1,000, for the local inference, for local transfer, it does not impact significantly our inference, but for the global transfer, when we simulate this up to 1,000 event, there is a huge variance, and obviously it does impact the inferences. So what we have reconstructed. So based on this method and the Bayesian framework, we reconstructed LUCA as a halotolerant hyperthermophile, although it is still, there is debate about the hyperthermophily of LUCA. It has evolved in neutral pH and in aquatic environments. It was anaerobic, chemolithotrophic, and curiously, it has catalase and oxidase, which suggests that it might, it was probably able to resist oxidative stress, but we know that concentration of oxygen at that time might have been like less than 0.001. So the presence of catalase and oxidase may be also due to the fact of possible ancient horizontal gene transfer. So the descent of other descent of LUCA, the last archaeal common ancestor, has also similar characteristics as LUCA, as being halotolerant, hyperthermophile. We were not sure if it was aerobic or anaerobic. For the last bacterial common ancestor, it was also anaerobic, chemolithotrophic. It has also catalase, and it was halotolerant and thermophile. It was not hyperthermophile, but thermophile, and evolved in neutral pH. So for now, anything that we think about LUCA, like for example, its morphology, it's based on intuition. So we think that it might have looked like a prokaryote, but it has never been reconstructed. So here, the variety of bacterial shape or archaeal shape is huge, but we simplified this into 3 main categories, like cocoid, that it just divide, and the rod, that it has a known elongation machinery, and ovoid, that has like a septal growth and then division. So when we reconstructed the ancestral state, we found that LUCA was probably a ovoid motile cell that has a monodermic, it was monoderm, like having one single membrane, and we were not sure it was spore forming or not. And it has a genome size of approximately 2.5, which is close to current genome size in some bacteria and archaea. So the archaeal common ancestor was also ovoid, motile, has a cell wall, and was monodermic. And the last bacterial common ancestor was likely a rod-shaped like bacterium. It has a cell wall. We were not sure it was monodermic, and it was probably non-spore forming. This reconstruction of the last bacterial common ancestor is similar to what has just been done recently by Coleman using a genomic approach, except that they found it was a didermic cell. So given this complexity of LUCA, shape and size and genome, it's given also the Earth formation timeline, and given when the LUCA had appeared on Earth at the first time, life must have been evolved very quickly, cellular into complex cells, and it was probably as much complex as many modern bacteria and archaea. So this is one of the message. The second message I want to tell you, and that is for me very important, since I worked with many biophysicists and mathematicians, is the scale at which we are observing or studying LUCA or microbes. So at the small scale, at the micro scale, the physics became counterintuitive. It's not the physics we are experiencing daily. Gravity has not the same importance that it has on macroscopic organisms. And by studying microbes and by reconstructing ancestral environment or ancestral selective pressure, even at the time of LUCA, we have to consider the microscale biophysics, like, for example, the low Reynolds numbers in which it evolves, Brownian motion, kinematic reversibility, and molecular diffusion, just to cite a few. So this is very important elements in when studying microbes. We tend generally, I highlight this because we tend generally as microbiologists to think with our intuitive physics when observing microbe, while we should not. Finally, if we look at life in space, I will confidently think that probably it will be microbial first before it can be if we find life in at least our solar system. I will look for microbials, and I will look for motility. I will look for DNA. I will look for any sign that is similar to what we observe on microbes on Earth. And with this, I would like to thank my colleagues and collaborators, Stuart, Say, Chris, and Andrew, and thanks if you have questions.
Great, thank you. So we have time for a few questions. On the left, or
Thank you very much for the
Nope.
presentation. Again, Vladimir Subbotin, University of Wisconsin. And you mentioned that there is no participation of gravity due to small size, right?
I said it's negligible. It's very negligible in comparison to our scale. Yeah.
So on a scale of a sphere of 1 micron, considering different in a density 10%, it's going to descend in the water with a speed 1 millimeter per hour. If it's a 5 micron, it's 1 millimeter, 1 micron is about 24 hours. And 5 microns radius is about 1 hour.
Yes,
So,
approximately.
Yeah. So it's still sizable moving.
It does. What I mean by that is it will not have really the same impact as it. Of course, it has. There is gravity at small scale, gravity at all scales, but it is not in terms of evolutionary impact or selective pressures. I don't think it will be comparable to what we observe on macro scale organisms.
Unless-
That's just what I mean.
Yeah, unless selective pressure is UV.
Yeah.
Yeah, thank you.
Next question.
Hi, that was a great talk.
Thank you.
Can you tell us about what you think that happened between in terms of the genomic increase between the last universal common ancestor and to the last bacterial common ancestor? Because it seems that you found like some increase in the genome, right? But not in the last archaeal common ancestor. If can
You
come
mean
back.
here?
Yeah, from 2.5 to. Do you think that was like new genes evolving, like or gene transfer, or doubling of like genomic duplication, something like this?
That's a good question. I personally, I don't have like a clear explanation for that. All what I can say is that the reconstructions were done independently. So we tested for trends if there is increase or decrease in genome through time using different phylogenies, but I didn't specifically test for like the increase or decrease from LUCA to LACA, for example. But thanks, that's a good question. Yeah.
So unfortunately, we're out of time.
Yes.
But let's thank our speakers again. And thank you everyone for your great questions and being here for our session. Enjoy the rest of the conference.